Pleasant day to each and everyone. So my name is Christinda and Ikabite, and I'm going to report about the MNS blood group system. But before we go to the topic, I'm gonna discuss first to you its history. So antigens MNN were first discovered and described by Karl Landsteiner and Philip Levine in 1927. So these two scientists immunized the rabbits in the human RBCs. That's why they found out antigens MNN. So another scientists were Walsh and Montgomery, wherein they discovered the S antigen that appeared to be genetically linked to MNN, and also its antithetical partner S was also discovered in the year 1951. Family studies demonstrated the close linkage between the genes controlling M and S. So here is an example of a table of prevalence of common M, N, and SS phenotypes. So in the first phenotype, M positive and negative, the whites have 28% while the blacks has 26%. The M positive and positive, 50% for whites, 44% for blacks. So in the third phenotype, the M negative and N positive has 22% in whites, while in blacks, they have 30%. In the fourth phenotype is the S positive, S negative. The whites has 11%, while the blacks has 3%. So in the S positive, 44% are whites and 28% are blacks. So in the S negative and S positive, there are 45% whites and 69% of blacks. In the last phenotype, the S negative, U negative, has only 0% in whites, while in blacks, less than 1%. So there you can see that there is a disequilibrium in the expression of S and S with M and N. So you can see in whites, the common haplotypes were calculated to appear in the following order of relative frequency. Like for example, NS is greater than MS, and then MS is greater than MS, and then MS is greater than NS. So you can see that there are differences in terms of each phenotypes. So next is the antibody to a high prevalence antigen, which is U. It is for almost universal distribution. So this was named by Weiner in the year 1953. So by the way, the complete name of Weiner is Alexander S. Weiner. So the MNS blood group system has been assigned by the ISBT number 002, second after the ABO. So like let's go back to the antibody to high prevalence antigen. So the observation of this from Greenwald and his 24 colleagues, that all URBCs were also connected with S negative antigens. So this resulted to the inclusions of U antigen into the system. So here are the summary of MNS antigens. So the first antigen is M. MNS1 is the ISBT number and polymorphic, and it is discovered in the year 1927. In the antigen N, it is MNS2. It is polymorphic, and it was also discovered in the year 1927. For the antigen S, It is MNS3. It is also polymorphic, and it was discovered in the year 1947. In the antigen small s, so it is MNS4. It is also polymorphic, and it was discovered in the year 1951. Antigen U, MNS5, it is high in prevalence. It was discovered in the year 1953. And then antigen HE, it is MNS6. It has low prevalence, and it was discovered in the year 1951. So next is the antigen MI raised A. So it is MNS7. It has a low prevalence and also discovered in the year 1951. The antigen MI raised C, it is MNS8. It has low prevalence and it was discovered in the year 1953. Antigen VW, it is MNS9. It has also a low prevalence and it was discovered in the year 1954. Antigen MUR or more, it is MNS10. It has low prevalence and it was discovered in the year 1961. Antigen MI raised G is MNS11, so it has low prevalence and it was discovered in the year 1958. Antigen VR is MNS12. It has also a low prevalence and it was also discovered in the year 1958. So another antigen is antigen M raised to E. So it is MNS13. It has low prevalence and also it was discovered in the year 1961. Antigen MT raised A is MNS14. It has also low prevalence, discovered in the year 1961. Antigen ST raised A, it is MNS15. It has low prevalence and it was discovered in the year 1962. RI raised A is MNS16. It has also a low prevalence. It was discovered in the year 1962 also. And then antigen CL raised A is MNS17. It has low prevalence and it was discovered in the year 1963. So the antigen NY raised A is MNS18. It has also a low prevalence and it was discovered in the year 1964. Next is antigen HUT or hot. It is MNS19. It has also a low prevalence. It is discovered in the year 1966. Another antigen is HIL, which is the MNS20. It has a low prevalence and it was discovered in the year 1966. Antigen M raised to V. It is MNS21. It has low prevalence and it was discovered in the year 1966. Antigen FAR is MNS22. It has low prevalence and it was discovered in the year 1968. Another antigen is small letter S raised to capital D. It is MNS23. It has low prevalence and it was discovered in the year 1978. Antigen MIT is MNS24. It has low prevalence and it was discovered in the year 1980. So another antigen is DANTO. So they are MNS25. It has low prevalence. It was discovered in the year 1982. Another is HOP. It is MNS26. It has low prevalence and discovered in the year 1977. So another is NOB. So it is MNS27. It has low prevalence. It is discovered in the year 1977 also. Antigen En raised to A is MNS28. It has high prevalence. It is also discovered in the year 1969. Antigen ENKT or MNS29 is high in prevalence and discovered in the year 1986. Antigen N, in closed quotation, is ISBT number MNS30. So it has also a high prevalence. It is also discovered in the year 1977. So another antigen is OR or OR. It is MNS31. It has low prevalence. It was discovered in the year 1964. Antigen DANE or DANE is MNS32. It has low prevalence. It was discovered in the year 1991. Another antigen is TSEN rather. It is MNS33. It has low prevalence and discovered in the year 1992. Antigen MINY is MNS34. It has low prevalence and it was discovered in the year 1992. Antigen MOT or MUT is MNS35. It has low prevalence and discovered also in the year 1992. Another antigen is SAT or SAT. It is MNS36. It has a low prevalence and it was also discovered in the year 1991. So another antigen is ERIK or ERIK. It is MNS37. It has low prevalence. It was discovered in the year 1993. Another antigen is EN raised to A. So it is MNS38. It has low prevalence. It was discovered in the year 1983. Antigen ENEP or ENEF, ENEP rather, it is MNS39. It has high prevalence. It was discovered in the year 1995. Antigen ENEH or MNS40 has high prevalence. It is discovered in the year 1993. Antigen HAG is MNS41. It has low prevalence and it was discovered in the year 1995. Another antigen is ENAV. It is MNS42. It is high in prevalence. It was discovered in the year 1996. Another set of antigen is MARS or MNS43. It has a low prevalence. It was discovered in the year 1992. Antigen ENDA is MNS44. It has a high prevalence. It was discovered in the year 2005. Another antigen is ENEV. It is MNS45. It has high prevalence. It was discovered in the year 2006. And lastly, MNTD. It is MNS46. It has low prevalence and it was discovered in the year 2006. So there are 46 antigens that have been included in the MNS system. So making it almost equal to RH in size and complexity. So most of these antigens are low prevalence and were discovered in the cases of HDFN or incompatible cross-match. So others are high prevalence antigens, and also the antibodies of these low and high prevalence antigens are not commonly encountered in the blood bank. So these genes encoding the MNS antigens are located on the chromosome 4. So next is the M and N antigens. So these MNR antigens recognize both amino acids and carbohydrate chains. They are antithetical and differ in their amino acid residues at positions 1 and 5. So they are found on a well-characterized glycoprotein called glycophorin A or GPA. So GPA is the major RBC sialic acid rich glycoprotein or the C-alkoglycoprotein or the SGP. So this, in terms of antithetical and differ in their amino acid residues at positions 1 and 5, and in terms of GPA, the antibody reactivity may also be dependent on adjacent carbohydrate chains. So these are rich in sialic acid. Also, there are about 106 copies of GPA per RBCs. So the antigens were well developed at birth. So you can see on the photo, in the glycophorin A, the MNN antigens, as what I've mentioned earlier, their difference is only their positions 1 and 5. While in the glycophorin B, there are antigens U, antigen S and S. So they also differ their sequence in the position 29. So because M and N are located at the outer end of GPA, so they are easily destroyed by the routine blood bank enzymes physin, papain, and bromelain, and also by the less common enzymes trypsin and pronase. So this figure shows a comparison of uh, glycophorin A and also glycophorin B. So you can see there are different um, lines in the graph shows different amino acids. So next is the antigens are also destroyed by ZZAP, which is a combination of DTT and papain or physin. But they are not affected by DTT alone. So there is the 2-amino ethiolysouronium bromide or the AET. There is also alpha chymotrypsin, chloroquine, or glycine acid at that treatment. So you can see on the picture, here is the chemical formula for DDT, and also the picture with a green and red spiral shapes. This is the papain and the other one is the physin. So MNN antibodies are heterogeneous. So some may recognize only specific amino acids, but others both amino acids and carbohydrate chains. So treating RBCs with neuraminidase, which cleaves sialic acid, which is also known as neuraminic acid or neonac. So they abolish the reactivity with only some examples of antibody. So because of antigen dosage, many examples of anti-M may react better with M positive and negative RBCs, wherein its genotype is MM, than with M positive and positive RBCs, wherein the genotype is MN. So the very weak anti-M may not react with M positive and positive RBCs at all. So making this an antibody identification difficult. So many examples of anti-M are naturally occurring silane agglutinins that react below 37 degrees Celsius. So although we may think of agglutinating anti-M as IgM, 50% to 80% are IgG or have an IgG component. So anti-M appears to be more common in children than in adults and is particularly common in patients with bacterial infections. And also, they do not bind complement regardless of their immunoglobulin class, and they do not react with enzyme-treated RBCs. So antibody reactivity can be enhanced by increasing the serum to cell ratio or incubation time, or both by decreasing incubation temperature or by adding potentiating medium such as albumin, low ionic strength saline solution or LISS, or polyethylene glycol or PEG. So as long as the anti-M does not react at 37 degrees Celsius, it is not clinically significant for transfusion and can be ignored. So when anti-M reacts at 37 degrees Celsius, it is sufficient to provide units that are cross-match compatible at 37 degrees Celsius. And also at the antiglobulin phase, without typing for M antigen, so the anti-M rarely causes HTRs, decreased red cell survivals, or HDFN. So some examples of anti-M are pH dependent. So they react best at pH 6.5. So these antibodies may be detected in plasma, so which is slightly acidic from the anticoagulant, but not in an acidified serum. So other examples of anti-M are react only with RBCs exposure. I mean RBCs exposed to glucose solutions. So such antibodies react with M-positive reagent RBCs or donor RBCs that are stored in preservative solutions containing glucose, but they do not react with freshly collected M-positive RBCs. So let's go to anti-N. So the serologic characteristics of the common anti-N made by individuals whose RBCs type M positive and negative and S positive or S positive are similar to those of anti-M. So a cold reactive IgM or IgG silane agglutinin that does not bind complement or react with enzyme-treated RBCs. And also, it can demonstrate dosage reacting better with M negative and positive, or its genotype is an N. So RBCs then with M positive and positive, or the genotype is MN, RBCs has also a rare examples that are pH or glucose dependent. So anti-N is not clinically significant unless it reacts at 37 degrees Celsius. So it has been implicated only with rare cases of mild HDFN. Then anti-N is less common than anti-M. So it is a potent anti-N that can be made by the rare individual whose RBCs type are M positive and negative because they lack both N and GPB that has the N activity. So next is the anti-N was also seen in renal patients. So regardless of their MN type who were dialyzed on equipment sterilized with formaldehyde, and also dialysis associated anti-N reacts with any N positive or N negative RBCs that are um, treated with formaldehyde and is called the anti-NF. So formaldehyde may alter the M and N antigens so that they are recognized as foreigners. So the antibody titer is the one decreases when dialysis treatment and exposure to formaldehyde stops. It is because anti-NF does not react at 37 degrees Celsius. It is clinically insignificant, especially for transfusion. So let's go to anti-S and anti-S, big letter S and small letter s. So most examples of anti-S and anti-S are IgG, which is reactive at 37 degrees Celsius, and the antiglobulin test. So a few express optimal reactivity between 10 degrees Celsius rather and 22 degrees Celsius. It is by silent indirect antiglobulin test. So if anti-S or anti-S specificity is suspected, but the pattern of reactivity is not clear, incubating test at room temperature and immediately performing the antiglobulin test. So this can be done without incubating at 37 degrees Celsius, wherein it can help to identify. So the antibodies may, in terms of the identification, so there is the dosage effect, which can be exhibited, by many examples of anti-S and anti-S, although it may not be as dramatic as seen with anti-M and anti-N. So this dosage effect is commonly seen in anti-S and anti-S antigen. So the antibodies may or may not react with enzyme-treated RBCs. So depending on the extent of treatment and the efficiency of the enzyme. So although seen less often than anti-M, anti-S, and anti-S, are more likely to be clinically significant. So they may bind complement, and they have been implicated in severe HTRs with hemoglobinuria. So they have also caused HDN. So these units selected for transfusion must be antigen negative and cross-match compatible. It's because only 11% of whites and 3% of blacks are small letter S negative. So it can be a lot difficult to provide blood for a patient with anti-S. So big letter S negative units are much easier to find because 45% of whites and 69% of blacks are S negative. So antibodies to low prevalence antigens are commonly found in reagent NTS. So this can cause uh, a discrepant antigen typing results. So let's go to the biochemistry. So GPA is the structure carrying the MNN antigens. It is a single pass membrane SGP that consists of 131 amino acids. Another is that the hydrophilic NH2 terminal end, which lies outside of the RBC membrane, has 72 amino acid residues. So 15O glycosidically linked oligosaccharide chains, or the GAINA serine or threonine, and 1N glycosidic chain, or the sugar asparagine. So the portion that traverses the membrane is the hydrophobic and contains 23 amino acids. So the hydrophilic COOH end, which contains 36 amino acids and no carbohydrates, so lies inside the membrane and also interacts weakly with the membrane cytoskeleton. So the GPB is the structure carrying the S and U antigens. So they consist of 72 amino acids and 11 O-linked oligosaccharide chains and no N-glycans. So it has an outer glycosylated portion of 44 amino acids and a hydrophobic portion of 20 amino acids that traverses the RBC membrane, and also a short cytoplasmic tail of 8 amino acids. So next is that the first 26 amino acids on JPB are identical to the first 26 amino acids of the N form of GPA or GPEN. So this N activity of GPB is denoted as N quotes, to distinguish it from the N activity of GPAN. So anti-N reagents are formulated to not recognize the weak reactivity of the N coats structure. So the U antigen expressed when normal GPB is present, and also is located very close to the RBC membrane. So you can see it on the U phenotype section. So next, before that, the most O-linked carbohydrate structures on GPA and GPB are branched tetrasaccharides containing 1 GAINAC, 1 GAL, and 2 neonac or sialic acid. So neonac helps give the RBC its negative charge. So about 70% of the RBC neonac is carried by GPA, and also about 60% is carried by the GPB. So GPA associates with protein bond 3, which affects the expression of the antigen WRB of the Diego blood group system located on the protein bond 3. So GPB appears to be associated with the RH protein and also the RH associated glycoprotein complex, which is an evidence by the greatly reduced S and S expression on the RH null RBCs. So other antigens within the MNS system have been evaluated biochemically and at the molecular level. So some are associated with altered GPA because of amino acid substitutions or changes in carbohydrate chains. So others are expressed on variants of GPA or GPB. So still others result from a genetic event that encodes a hybrid glycophorin that has parts of both GPA and GPB. So the altered glycophorins are associated with changes in glycosylation. So changes in molecular weight, loss of high prevalence antigens, or the appearance of novel low prevalence antigens. Or there is an alterations in the expressions of MNS antigens. So some RBCs that lack GPA or GPB are not associated with disease or decreased RBC survival. So it is incorporated to the disease associations, but GPA and GPB are expressed on renal endothelium and epithelium. So in terms of genetics, the genes GYPA and GYPB, which is a code for GPA and GPB respectively, are located on the chromosome 4 at 4q28-q31. So the known alleles for GYPA is the genotype MN, and GYPB is the genotype, antigens S are codominant. So the genes are highly homologous, meaning they are very similar and probably arose by gene duplication. So GYPA is considered to be the ancestral gene. So GYPA is organized into 7 exons. GYPB has a size and arrangement similar to those of GYPA, but has only 5 coding exons plus 1 non-coding or pseudoexon. So a third highly homologous gene is the GYPE. So this does not appear to make a glycoprotein that has been definitively recognized on the RBC surface, but it participates in gene rearrangements that result in variant alleles. So there is a misalignment of GYPA and GYPB during meiosis. So followed by an unequal crossing over appears to explain some of the variant glycophorins rather observed in the MNS system. So the resulting new reciprocal GYP A A-B and GYP B-A genes encode GYPA encodes GP A-B and GPB-A hybrid glycophorins. So the point of fusion between the GPA and the GPB is in the part of the hybrid glycophorin that can give rise to novel antigens, like for example, antigen of low prevalence. So more complex variant glycophorins, so a gene conversion event probably occurs. So gene conversion involves a non-reciprocal exchange of genetic material from one gene to another homologous gene. So as with the hybrid are resulting from crossing over, so the new amino acid sequence at the junction of the hybrid glycophorin can give rise to novel antigens of low prevalence. And also it is expected that antigens may be missing if the coding exons are placed by the inserted genetic material. So next is the GPA and GPB deficient phenotype. So RBCs of 3 rare phenotypes lack GPA or GPB or both GPA and GPB. So consequently, they lack all MNS antigens that are normally expressed on those structures. So the RBCs usually type S negative, U negative. And these individuals can make anti-U in response to transfusion or pregnancy. So anti-U is typically IgG and has been reported to cause severe and fatal HTRs and HTFN. So the U antigen is resistant to enzyme treatment. So thus, most examples of anti-U react equally well with untreated and enzyme-treated RBCs. However, um, there are rare examples of broadly reactive anti-U that do not react with papain-treated RBCs. So after RBCs, um, in terms of RBCs, rather, so this high prevalence antigen is found on the RBCs of all individuals. So except for those of African Americans, because 1% to 35% are Africans. So they lack GPB because of a partial or complete deletion of GYPB. So here, some examples of anti-U react with apparent URBCs, although weakly but adsorption and elution. So such RBCs are said to be U variant. So this have an altered GPB that does not express S or S. So there is a strong correlation between the low prevalence antigens, like for example, the antigen HE, which is found in about 3% of the African Americans, and U variant expression. So because examples of anti-U are heterogeneous, U units selected for transfusion must be cross-matched to determine compatibility. So some patients may tolerate U variant units, others may not. So many examples of anti-U are actually anti-U plus anti-GPB. So if the patient is U negative and N negative, so the antibody may actually be a potent for anti-N plus anti-U. So this makes the search for compatible blood more difficult. So next is the EN raised to A negative phenotype. So in 1969, Darnborough and co-workers and Forogel and colleagues describe an antibody to the same high prevalence antigen, which is called EN raised A for envelope. So which reacted with all RBCs except those of the propositee. So in both of these cases, the EN A negative raised A negative individuals appeared to be M negative and negative with reduced to neonac on their RBC. So the RBCs of the two individuals were mutually compatible. So most EN raised A negative is individuals produce anti-ENA, which is an umbrella term for reactivity against various portions of GPA unrelated to M or N. So, but not all antibodies detect the same portion. So, anti-ENA TS recognizes a trypsin-sensitive area on GPA between amino acids with in 20 and 39. So, anti-EN raised A FS reacts with a friction sensitive area between amino acids and 56. So anti-ENEFR reacts with a fissin resistant area around amino acid 62 to 72.1. So although the gene responsible for this phenotype has been termed EN, wherein it is now known as EN raised A negative phenotype, has more than one origin. So most often the EN raised A negative phenotype results from homozygosity So it is a rare gene deletion at the GYPA locus. So consequently, no GPA is produced, but GPB is not affected. So this type of antigen inheritance is called EN raised A negative fin. So representing the type uh, described in the Finnish report. So however, this antigen phenotype in the English report is probably represents heterozygosity for a hybrid gene along with a very rare MK gene. So this type is often called EN raised A negative UK. So several other EN raised A negative phenotypes have been reported that result from homozygosity for a variant allele or inheritance of two dissimilar variant alleles. So anti-EN raised A can be confused with two other specificities that react with all normal RBCs, the anti-PR and the anti-WRB. So anti-PR does not react with enzyme-treated RBCs and can be confused with anti-EN raised AFS, while anti-WRB does not react with EN raised A negative RBCs, but does react with enzyme-treated RBCs and can be confused with an anti-EN raised AFR. So anti-EN raised A has caused severe HTRs and HDFN. So it is extremely difficult and maybe impossible to find units compatible for patients with anti-EN raised A siblings for wherein they are a potential source of compatible blood if they are also ABO and RH compatible. So next is the MK phenotype, M raised to K phenotype. So the rare silent gene MK was named by Metataxas and Metataxas Boller in 1964 when they found an allele that did not produce M or N. So a second family showed that MK was also silent at the SS locus. So several other MK heterozygotes have been found. So in 1979, two related MK blood donors were found in Japan. So the RBCs of these individuals type M negative, N negative, S negative, S negative, U negative, EN raised A negative, WR raised A negative, B negative, but they had a normal hematologic picture. Next is more individuals have since been identified, and it is now known that the MK gene represents a single near complete deletion of both GYPA and GYPB7. So thus MK is the null phenotype in the MNS system. So MK genotype is associated with decreased RBC sialic acid content but increased glycosylation of RBC membrane bands 3 and 4.1. So other antibodies in the MNS system, antibodies to antigen other than MNS and S are rarely encountered and can usually be grouped into two categories. So those directed against low prevalence antigens and those directed against high prevalence antigens. So antibodies to high prevalence antigens are easily detected with antibody detection RBCs. So antibodies to low prevalence antigens are rarely detected by the antibody detection test but are seen as an unexpected incompatible cross-match or an explained case of HDFN. So few hospital blood banks have the test cells available to identify the specificity, but enzyme reactivity and MNS antigen typing may offer clues. So when antibodies to low prevalence antigens are encountered, it is common practice to transfuse units that are cross-match compatible at 37 degrees Celsius and also in the antiglobulin phase. So typing sera for MNS antigens other than M and N and S and S are not generally available. So the antigen status of compatible RBCs can seldom be confirmed. So also, when one antibody to a low prevalence antigen is found, antibodies to a low um, prevalence antigens are also frequently present in the same serum. So if the antibody is directed to a high prevalence antigen, the assistance of an immunohematology reference laboratory is generally needed to identify the antibody and obtain appropriate antigen negative units. So next is the autoantibodies. So autoantibodies to MNN have been reported. So not all examples of anti-M in M-positive individuals or anti-N in N-positive individuals are autoantibodies. So many fail to react with the patient's own RBCs. So it may be that these individuals have altered GPA and that their antibody is specific for a portion of the common antigen they lack. So autoantibodies to U and EN raised A are more common and may be associated with warm type autoimmune hemolytic anemia. And lastly is the disease association. So GPAM may serve as the receptor by which certain pyelonephritogenic strains of E. coli gain entry to the urinary tract. And also, the malaria parasite Plasmodium falciparum appears to use alternative receptors, including GPA and GPB, for cell invasion, and some of these receptors are also involved in UNAC. So that's all of my report. I hope that I have discussed it thoroughly. Thank you.